Great. Good afternoon and welcome to our webinar that we organized from the SbD4Nano project. Today's topic is a deep dive into the project's tools and e-infrastructure through a real case study. And we have prepared a very nice program for the next 2 hours with very interesting experts' presentation. And quickly for the housekeeping rules. So just that you know, as participants, you will be muted during the presentations, and we will allow you to speak up in the Q&A session at the end of the webinar after the presentations. At any time, you can post questions or comments in the chat. So I will try to monitor the chat while the presentations are running, and you will all receive also the recording of this webinar via email afterwards. So my name is Susanne Resch. I work for BNN, and I'm a work package leader for stakeholder engagement, dissemination and exploitation in the SbD4Nano project. And I'm very happy to guide you a little bit through today's webinar. And I think we can already start with our first presentation. So we start with the project coordinator, Carlos Fito from Itene. And he will talk about our project SbD4Nano, new solutions to support the development of safe and sustainable by design nanomaterials. So Carlos, I give you the floor for your presentation.
Your mic is on now, Carlos.
Actually, we don't hear you yet.
Okay.
Now.
Now, yes. Thank you, Susanne, for the presentation. Also, thank you all the attendees just to have time to be here. This is one of our events that we want to organize just to present what is going on in this project in SbD4Nano. As you know, SbD4Nano is a project that is related with the big framework of the Safe by Design. In this case, other projects at the moment are also dealing with the sustainability, but our project was more focused on the safety part of this kind of context. So I will try in this time that I have here in this webinar to present our project, an overview of where we are at the moment. Or of course, we are approaching to the end of the project, so we will have today the opportunity of having a big picture of the big outcome from the project. Also, we have here work package leaders that will present you an overview of the main results. I hope that you enjoy during this time, and of course, we will be open to any question. And also, you're welcome to participate in other webinars that we will organize in the project, and also in March. Also, we have a big event in Austria where you will have the opportunity to also see more about our projects. So next, please, Stella. So the motivation of the project, so that's very important because you know that SbD4Nano is related with the Safe by Design. In this case, you can see that the title of the project start with the word computing infrastructure. So that means that our main outcome here is a kind of big tool, in this case, a software tool that is developed just for the needs of the industry. So that's very relevant. So we are focusing on defining different solutions for the industry. Itene is the project coordinator. As I said before, we are approaching to the end of the project, scheduled in March 2024. We have a lot of partners here in this project, up to 23 partners, but the most relevant thing is that we have up to 9 companies, small and medium enterprises, but also large companies. And as I said, our main objective is to develop a new infrastructure in terms of software and tools that are oriented just to support companies on the definition of new materials and safer products and also safer processes. You can see in the screen that we have different well-known organizations around the nanosafety area, like BioNanoNet or Jordas, TNO, also the Maastricht University, and also a lot of companies that we have here. Of course, Sunum, as a Turkish company, is developing a lot of activities in toxicology. So next, please. So in this case, as I said before, we have a big motivation that's support companies on developing safer materials and safer processes. So we know that there is a big lack of knowledge related with how to deal with this concept. So there are a lot of barriers to support the implementation of the safe by design concept. So for one side, there is a lack of knowledge considering the toxicological properties. Also, there is a big lack of knowledge related with the potential exposure to nanomaterials in the workplaces, but also there is a lack of data or tools related with the implementation of the concept. There is a few limited tools like decision support system or risk assessment tools that are really focused on helping or supporting the industries on organizing the data to implement this content. So next, please. So in terms of these barriers, so our project is really focused on trying to overcome these barriers. So we have barriers related with the lack of knowledge, as I say, related with the toxicology, how we can analyze the toxicology of a material if we don't have real access to the data. Of course, we need to come up with solutions that are safer but also functional. So there is also a lack of resources to analyze how the functionality, for example, is being modified by introducing, for example, new surface modifiers on the surface. So that's also critical. We can develop safer products, but always this kind of product has to be functional. Also, there are some guidance at the moment from the European Commission, for example, but there are some, limitations related with the applicability of the guidelines to the real companies, the small company that is dealing with the material. So there is a barrier related with the lack of this kind of guidelines. So in our project, we are going to develop, or we are developing this kind of guidelines. Also regulation. So we need to inform regulators about potential outcomes from our case studies. And we can support regulators on defining good policies regarding safe by design. And also it's important in the product value chain, a big communication. So we have to support communication between the raw material producers, the people that is producing the nanomaterials, and the ones that are using nanomaterial, for example, paint formulators or manufacturers of plastic materials. So we need to support and make more dynamic this communication. That's one of the key objectives of the project. So don't make these things happen in the project. So all these barriers are for SMEs, but also for large companies, and are very relevant. So our project is more or less focused on trying to overcome this kind of barriers. So next, please. So in this case, how we deal with that. So our objective is to support all these barriers by developing solutions. So, and these solutions at the end are software tools, databases, modeling, but also experience from case studies. And we have compiled all these kind of solutions in a kind of big structure, and the name of the structure is the e-infrastructure that I will present, only an overview, and my colleagues from ThinkWorks, they will present, the whole picture of the tool and how the tool can support you in overcoming all these barriers. So next, please. You will see here, a first presentation of this infrastructure. At the end, the concept of the project was, of course, develop this kind of tool, but we were always thinking in four main pillars. For one side, support companies on calculating the severity in terms of hazard. Also, we need to support the company on a better knowledge of the potential exposure of a process dealing with nanomaterials. So that's the second pillar, the exposure. Always we have the third pillar, that is the cost, which is the cost of implementing, for example, modifications in the company to reduce the exposure, or which is the cost of reducing the potential toxicity of a material, or just modify the material or substitute the material for other. And finally, the fourth pillar is the performance, the functionality. So in our project, we merge all together. We have the stability, the exposure, the cost, and the functionality, the product performance, to come up with an index. It's the SbD index that will support the company for identifying proper solutions to support, the development of these safer processes and safer products, but always considering cost and functionality. That's very important. Next, please. You will see here a first picture of our infrastructure. Our infrastructure is based on three main parts. You can see that we have what we say SbD scenario assessment. So that's the real tool that Ralph from ThinkWorks will present you. This is the tool that is based in different kind of data, and will support the companies on selecting proper approaches and to reduce the toxicity or to reduce the exposure, but also identify the impact on the functionality. So that's very important. That's clear, and that's very important for us in the project. Also, we have SbD resources, and these SbD resources include, for example, a list of different tools. So we have a kind of inventory of different tools, guidelines, literature that is there, that is always important for the companies. Also, we have an instance for the nanomaterial database, so we can extract data from the nanomaterial. So that's also important in terms of toxicology. Also, we have the Athena. Athena is a kind of tool that is developed by the European University in Tarragona, and come up. It's important because the tool is intended to retrieve information from literature with data mining approaches. So you will have a lot of data from the scientific literature on potential SbD approaches and information for implement SbD approach. Also, we have a tool related with the regulatory requirements. It's a tool developed by Gene and support you for identifying regulatory requirements for your specific products. And also we have a connection with the TiO2 Diamonds platform, where you will find different kind of tools, like for example, tools like the nano exposure quantifier to compare the exposure in different processes. That's very important to design safer processes. That's very key, for example, for us in our project. So next, please. So, but always, we need to validate. So in our case, you will have this big infrastructure, but also we need to validate all the outcomes from the project. So to the same, we have up to 8 case studies that are very important for us in our project. And we want to validate, for example, the hazard profiling of our project, also the modeling framework related with exposure. Also, we have been working a lot with the companies in terms of the cost benefit. So we have been trying to know the potential cost of implementing this kind of approaches, like modifying, for example, a product or modifying something in the processes that is intended to reduce the exposure. So that's very key. So in the next slide, you will see more details on the potential case studies. So next, please. So here you can see our core. This is our core in our project. So we have different companies. Of course, these companies are producing different kind of materials. For example, graphene oxide or titanium dioxide or thin oxide. All these companies are producing these products because these products have an application, and this application is based on the market. So the market is demanding products that contains nanomaterials, and now implementing the safe by design concept. Also, the market and the European policies are demanding safer products, but always considering the safety of workers. So the companies, they know that it's important to reduce several issues related with the toxicology, and we start working with the companies to try to reduce some of these issues. In the next slides, please, you will see more details on some of the case studies. For example, Ambrosia Lab, that is a company working for the cosmetic industry. So they have titanium dioxide and tin oxide. And in our project, we have been working in different properties, modifying, for example, the structure of the materials by Sunum. And at the end, we find safer products, with less toxicity. So in the next slide, please. Also, we have Creative Nano, is a Greek company also producing titanium dioxide. Also, they have been working with us in the project in 2 areas. We have been developing new paints that are safer by design. So at the end, we define some modifications of the structure to reduce the toxicity, but also the dustiness. So that's also very important because we reduce the exposure in the workplaces. So next, please. In the case of Avanzale, they are producing graphene oxide materials, and we also find a way to reduce the toxicity of several materials that they are producing. But also we have implemented several strategies to reduce the potential exposure in workplaces. So that's also very important because they have not lost any functionality at the end in the final product. The next, please. So considering Apply Nano, also they are producing graphene oxide, but in this case for medical application, for drug delivery and biomedical application. And also we find a way to reduce the potential exposure during the production of this kind of materials. And of course, we make more biocompatible the material using different kind of surface modifiers. That was very important also in the life of the project. So next, please. So in the case of Group Antolin, they are producing material for the automotive industry. And they were also worried about the potential exposure. And also we find the way to reduce the exposure in the workplace by introducing new approaches to control the exposure. And of course, also with Mustafa, with Sunum, we find also modifiers to reduce toxicity. That's also very, very key in the life of the project. So next, please. In the case of Axon, also they are a company producing different kind of materials, in this case more metal or based material for also biomedical application. And we test a lot of properties of the materials, and we come up with different recommendations to reduce potential hazard issues for the real application. That was also very, very important in the life of the project, and also we estimate potential modifications in the functionality. Next, please. So concerning Haiku Nano, also they are developing different material like silica nanoparticles, and we identify also different kind of products, surface modifiers to reduce the potential impacts on the toxicity, but also respecting, the functionality. And also they are now working with the scale up of the materials to produce in the market. And next, please. And also Laurentia, that's a Spanish company close to Aitene. We have here a very good sample. So they managed to reduce the toxicity by substituting some of the material that they use in the beginning of the project as sulfate agents. So they modify these sulfate agents, and now they decrease a lot, the potential impact. And of course, we have deal with not reducing the functional part of the project. So that's very, very key. And also next, please. And concerning Nanovector, also they are more working in the pharma, so they have other kind of things. So we have here more activities related with exposure in the workplace. So trying to manage, risk management measures in different activities, and also they are very interested in trying to manage potential costs using the tool. And next, please. In conclusion, at the end, the important thing here is that using all these case studies, we have learned a lot of things from the companies, So what we know at the moment is very important to consider, reduction of the toxicity, reduction of the exposure, but always we need to know the potential influence on the functionality and the cost. So we make a lot of analysis and we come up with an influence diagram. So we have identified critical factors that influence on the functionality or influence in the analysis, in the cost analysis. So at the end, the tool will give you an overview of which parameters will influence your functionality and your cost, and that's critical for your decision making. So that's more or less the final outcome of the project. So we know better these parameters, and all this now automated in our tool. And I think that Ralph will tell you more about this tool, and I hope you to enjoy this presentation from ThinkWorks, because you will see all the approaches that we have implemented. And next, please. Just to take home, just to close the presentation, I think that we know that there are lack of data, lack of guidelines, and all these things. But all these projects, in this case, SbD4Nano, I think that come up with solutions and tools that will support your company on implementing this big content, Safe by Design. So next, please. And finally, I think that you will be able to use the tool in a couple of months, by March, more or less. And you will have the library of approaches. Also, you will have resources to identify regulation, new guidelines to implement the concept. And of course, you are invited to follow the project, and in the future to see next events where we have the opportunity to present our key outcomes. So this is the one, I think the last one. Next, please, Stella. So just to close, I think that is validation and demonstration will be very clear. I think that also we will invite you as people that is here to participate in the validation phase of the project. But for us, it's very important that people can test all these kind of tools in the life of the project to support us, in improving our results. So I think that's the last one. So I hope that you have enjoyed this big picture of the project. You have here my contact, also the contact of my colleague Elena. And also thank you for the attention.
Great. Thank you very much, Carlos, for your presentation and for setting the scene and giving us the overview of the project, how we approach it, the motivation, and also the industrial case studies that we have. I don't see any urgent questions in the chat, so I would say we can directly move on with our next speaker, Mustafa Kula from Suno, and he will talk about the role of the surface chemistry and in vitro toxicity screening on safe byproduct design strategies. So Mustafa, we are very happy to have you as speaker now, and the floor is yours. You are still muted, but I guess we can solve this soon.
Yeah, okay, good.
You should be okay now.
Yeah, good, thank you. Well, thank you very much for this nice introduction, Carlos and Suzanne. And what I am going to do today is that as the institution that are in charge of work package 3, I'm going to tell you our effort to modify the surface of these nanomaterials, which are practically used in industry, and to see how the surface chemistry affects their toxicity, or the goal is to reduce their adverse effects through surface chemistry, of course. And then do the testing, in vitro screening, and then see how the in vitro screening is a valuable tool to understand their toxicity. Here's the outline of my talk. I don't know why this is going faster, but first I'm going to show you nanomaterials and their properties, and I'll give you some brief information about their safety. Then I will mention briefly about the safe by design concept. then the nanomaterial toxicity and the surface chemistry relation, then of course I need to show you some case studies, some materials, and how surface chemistry is affecting their behavior in biological system and environment. Then these are basically, we are doing two types of in vitro tests. The first group is, to check with the eukaryotic cells, related to human health. The other is the environmental concern, and that is, it's called ecotoxicity issues, and I will conclude my talk. Well, I think, the audience and everyone knows who are in this field that there is a vast majority of the vast number of nanomaterials available to us. Any material, technically can be brought into nano size to explore their function. Okay, so I'm not going to go through this, but if you are in this field, or if you are even related to this field, and I'm sure you know the diversity there. There's a huge number of particles out there. Okay, for example, this slide is just showing some TEM, SEM images from our studies, and then as you see, they are even available for a variety of different applications because of their functions, because they show unique properties in new, maybe novel products that might be useful for us. And this is the list of nanomaterials that we studied from the companies who are part of this project. Okay, as you see, I think Carlos nicely mentioned all these companies and their materials, and the nanomaterials we used from them, from their production. And as you see, it's just, again, it's a huge diversity here, different forms, different applications, and, and the different sizes and so forth. Okay, so, because I'm just trying to tell you that there is a huge variation in the nanomaterials. Okay, so what is the concern here? Concern is because if these materials are used in any application, at one point, of course, from the humans will be affected. Also, next, environment will be affected, because we first, as humans, we use a product, then where these materials go, they go to environment, right? So there are a lot of different scenarios, they can affect health and environment. So, for example, in biological systems, once they make into the human body, they may go through many different barriers, and they may damage the organs. And once they make into the cells, they can actually affect the cellular events that, resulting in different, situations, like including even cancer. So we need to understand how these materials behave in these biological systems. Okay, here's the next. The issue is that because can we make these materials safer? Okay, because we are using them, right? Okay, then we know that they are useful. Can we make them safer? So that is the whole idea. This safe by design idea is not new, but its application in nanotechnology is new. So actually, this is the definition of safe by design is to start all the safety concerns from the scratch during the R&D stage, then go through all the steps from the R&D stage to the release into environment. So for example, if you build a building, so you need to take care of all the concerns, because the safety concerns, because people will be going into the building, living in the building, right? We don't want to collapse them in 2 days later. So you need to take all the precautions before you start making the building. So the same idea here, because once we start, considering all the function, okay, why we are aiming to use, in the application, we need to start from the scratch, from the design stage, to make this material safer, and even after their use in the application. So, well, probably, I'm assuming that you know some about because if you are listening to this webinar, you know nanomaterials. Nanomaterials, from chemistry perspective, it is not that easy. If you're designing something, it's, for example, carbon nanomaterials. You make them, right? I mean, there's not much control until you make them. So the size, maybe you have a control. I mean, the shape, maybe I mean, these are difficult, things, the shape and size, for example, to control. So because you make them, because they are nice materials and they have nice functions, for example, electrical or some, carrier functions or such. So because you cannot play with their size and shape too much, because they're a carbon-based material, you just make them. So at the end, I mean, this isn't just one example, but if you consider other nanomaterials, you don't have much control, right? Because even you change the size, you change the function. So it is not much room there to play with their size and shape to make it safer. So what happens is that, here's the deal, because the idea is to change the surface chemistry, because this might help the interaction of these materials with the environment wherever they are residing in the matrix, which is, in this case, it's biological and biological medium, biological environment here, right? Because this is the question to why is because surface is the interface to the outside, mainly this, the biological medium. Okay, so, well, I mean, once you try to change the surface chemistry, you need to take, you consider a lot of parameters together. So which surface chemistry you're going to use, which molecules you will be using, what polymers you will be using. So then are they biocompatible, okay? Are they, once you make the surface modification, are they stable in the formulation? And then is it chemically suitable for that kind of application by considering your application? How about the cost? It's really important, right? Because if you're going to make a mass production, is it really possible, to use, possible to obtain it in a low cost? Because it's going to cost you, if a product is going to cost more, you don't want to use it, because no one is going to buy it, right? Also, you need to consider the toxicity concerns. For example, what is going to happen to this surface chemistry? Is it going to be stable on the surface? Is it going to be leaving, or if once it leaves, it is going to be toxic or not? And there are a lot of issues. If, for example, aggregation is another problem. Okay, after taking care of all these, concerns, now you need to consider the surface modification. So what happens is that surface modification is another issue, because you are going to again use chemicals, right? So you need to pick the best strategy to not to contaminate your nanomaterials, because all chemicals that are used in this modification strategy can affect the toxicity. Okay, are you going to use just a simple physical coating, or just attach the molecules that you selected through certain chemistry? Is this chemistry available? And all these, the question should be answered to develop a strategy, modification strategy. So now I'm switching to the point that how we handle this. From the our experience, and also from the literature studies. And what we did is, I came to the conclusion that small molecules, biological molecules, okay, with non-toxic biological molecules such as glycine and glucose, and these monosaccharides and then amino acids, and they're good candidates because they will not alter the function of nanomaterials as well. Because just changing the surface without changing too much of the other materials, I mean properties of materials. Okay, so then next step was to modify them. Today I'm going to show you especially these carbon-based nanomaterials later on with the in vitro toxicity testing. But here this is showing how carbon-based nanomaterials are modified, the chemistry and their modification strategies. But I included ethylene diamine to see if I change the surface, because if your glycine makes the surface negatively charged, ethyl-diamine makes the surface positively charged, and the glucose is just, of course, it's neutral. To see these effects that I made the changes later on, I'm going to focus on this. But before I go on, Carlos nicely presented all the companies who provided diverse nanomaterials with diverse surface chemistries. So for diverse applications, okay? It is not that easy, because I'm trying to make a point here later on when I start talking about the in vitro toxicity. I will, come back to this point, because the diversity is important. Of course, as I said, the function of the nanomaterials are really important as it comes to the modification, everything, all these things that I talked about, is really, the important not to change the function of the nanomaterial, because in the application at the end of the day, you need to use this in the product, and you want that your product is going to be functioning the same. So this is an example. For example, this is sulfur-doped titanium silicon oxide nanoparticles used by Creative Nano in Greece. They use these materials for photocatalytic paint, for example. Okay, so now I'm going to go into this nanotoxicity the tests. Okay, there are two in vitro tests. These are done in the laboratory. Okay, so the first is concerning the human health. The other is the environment. This is I'm going to just show you which tests we use in this project to concerning the human health. These are in viability tests, VST8 assay cell viability test we used, and the other test we used was the COMET assay for DNA damage. And the for environmental tests, it's just a little bit complicated, and we added more types of different tests that I'm going to, so ZSA and alga growth inhibition test, and earthworm acute toxicity test, and bioaccumulation tests. Then I'm going to talk about this, possible issues there as I move on, and to tell you the complete story here. Okay, here's the question. Maybe you may be thinking, okay, we are doing these in vitro tests. Okay, then we are concerned about the environment. Of course, a different story, but the human health is in the real, biological system in the human body. Are they going to be the same, their behaviors and their effects? There's an effort to correlate the in vitro and in vivo tests. Okay, so I mean, it is. Sometimes there's a good correlation, sometimes not. Because why this is so? Because nanomaterials behavior in the biological systems is truly, I mean, how they behave in a matrix truly depends on the matrix. Okay, if you change the environment, for example, if you have a PBS buffer, if you put these materials into cell culture medium, it behaves differently. If you throw them on the soil, it's just a different story. So that's why, if, for example, cellular study is just, it's just more maybe compatible. I mean, there's a correlation with this, in vitro and in vivo. Okay, there are some, literatures that are arguing that, it is possible or not possible, but there's some good correlation that I would say. The other point is because I told you about the diversity of these nanomaterials, because you cannot test them in vivo, all these diverse nanomaterials out there. So all the way to test them in the laboratory through high throughput testing. Okay, so then, of course, the cost and time, because you cannot just go and try on animals, because it's gonna cost you more and it's gonna cost you more time. So there's an effort to move from in vitro, I mean, to develop in vitro tests which mimics the in vivo systems, such as 3D cell cultures, and then also some microfluidic system and so forth. So that's why it is, in this project, we focus on in vitro. We don't want to, because we don't have the capacity to test that diverse nanomaterials in vivo testing. So now I'm going to show you some results with the in vitro toxicity testing. Just we selected 3 cell cultures. These cell cultures, first of them HSF is human skin fibroblast tissues, cells, and the other is human bronchial epithelial cells, and the other is the macrophage. Why we selected these cell lines? Because considering the exposure, nanomaterial exposure, how people are exposed to these nanomaterials. For example, if someone is making these nanomaterials, they probably exposed to these materials through the skin, or they can, airway, they can, breathe these materials. Then of course, once these nanomaterials make into the human body, what happens is that they, cause an inflammation and immune response. That's why we selected these 3 types of cell types. Then here's the results. For example, I show you the sulfur-doped titanium oxide, silicon oxide nanomaterials. As you see here, this material is just typical. Very typical. And with the in vitro testing, they show some toxicity, some don't show, depending on the sulfur content in these nanomaterials, because that is the concern. Because we usually know that titanium and silicon nanoparticles, they are not very toxic. They're okay, and even they are studied for drug delivery applications. But if sulfur is doped into it, so what happens? Do they cause problems for example, when they interact with cells? It is quite, showing that it is not really toxic to these cells with the time and concentration dependent study. This another the nanomaterials from Laurentio Technologies. We tested the silicon oxide nanoparticles on the surface. In one type of particle, there's only hydroxyl groups. In the second type is amino groups. This, the bottom of the slide is showing their characterization. Of course, we had to characterize fully to make sure that this material is really what it is meant. So as you see here in the different cell lines, it's obviously silicon oxide nanoparticles in this case is showing toxicity on these cell lines we tested. Because as I said earlier, why silicon oxide nanoparticles are not toxic, but in this case it is toxic because I told you there's a huge variation from synthesis. Even synthesis makes a big difference because who knows what kind of chemicals they used is causing this toxicity. Is it just the nanomaterial itself or other chemicals along with the synthesis? There's another study here is from group Antolin. They provided this carbon nanofibers. As you see here, you can see the TM images and SEM images, and there are, it's just really huge variation in size and length and so forth. And then it was my really interest to see how surface chemistry is gonna affect the behavior of these nanomaterials in the test tube. With the interaction with the cells. It is amazingly this change in surface chemistry. For example, glycine, and as you see from the graphs, modification with glycine and amine and glucose, and they all improve the toxicity of these materials, even though they are mass. But when we tested with the genotoxicity, it looks like that they are not, they are not very toxic. They are not really causing too much trouble for DNA damage. The another one is the graphene oxide nanomaterials, graphene oxides from Upline Nano Solutions. The similar story here as well. Then also the Avanzare. Okay, this is obviously. We see, although we see a trend in the how toxicity improves with surface chemistry, there is a variation in the response. Finally, in ecotoxicity tests, there is again a lot of data here and a lot of different types of tests. And I'm not going to go through all of these the tests and results, but I'm going to show you just a few of them. For example, zebrafish embryo toxicity testing. And then, of course, here during the testing, the stability of the testing medium is really important. First, the Italy and INL, they studied the behavior of these nanomaterials, modified nanomaterials in different mediums like fresh water and ultra pure water and so forth. Then they tested these animals and they found that there are some variations again in the results, and the glucose modified ones show more toxic behavior here for the Z test, the influence on the, as you see here on the table, you can see a lot of different, parameters are checked. And then this is something that is, interesting. And also the other test, for example, ALDA growth inhibition test, it obviously modified ones showing some effect on this case. This is interestingly. I mean, then because when it is compared to graphene oxide, but in Dafina acute immobilization test, they also, modified ones are making some impact on this toxicity, the picture landscape. But there is because I can tell you why this is so. Okay. Then also other tests, earthworm acute toxicity test and the bioaccumulation tests. So I mean, this is results are here is interesting because this the surface modification causing some effect, sometimes generating more toxic materials, sometimes generating less toxic materials. Actually, this is the reason why surface chemistry is an important point here to explore and understand, and along with the in vitro testing, because it provides a lot of data, okay, and can be used in the further studies. For example, in this project, work package 3 is concerned with the generating data from these testings and surface modifications and testing, and also literature, the data. Okay, in conclusion, I'm not going to, I think I'm almost done here, but it shows that surface chemistry is an important parameter to understand the how these materials behave. Okay, it is possible, very possible, to make nanomaterials less toxic nanomaterials using surface chemistry, and the diversity that is observed with the results is there are many different reasons for this. It could be nanomaterial itself, or it could be just the surface chemistry or the metrics these tests are performed. Okay, I mean, in general, I mean, we see that the surface chemistry is a critical parameter to define the behavior of these nanomaterials in biological systems. And this in vitro testing is really, really important. And there is I mean, I would say, in some cases, a good correlation with the in vitro and in vivo results. Then I'm going to conclude my talk, and thank you very much for your time.
Great. Thanks a lot for these insights, Mustafa. A lot of information and interesting topics. Thanks a lot. I don't see a question in the chat right now, but you will stay with us until the end also for the Q&A session. So I think we can also discuss later. So I will move on in the agenda and present our next speaker, Wouter Fransman from TNO. And after the safe by material design, we move in the topic of safe by process design to prevent exposure in the value chain. And with this, Wouter, I give you the floor. And yeah, thank you very much for presenting now.
Thank you very much, Susanna. And yes, as you said, we're now diving into the process design, which may be a new term for many of you, because when we speak about safe by design, usually people are dedicated to trying to change the material. But if you click to the next slide, I will show you that actually risk is a combination of hazard and exposure. And if we present it as being, let's say, the lion being a hazardous animal, and you expose yourself to that animal, there will be a risk. But the fact that risk is a component of two different elements, we also feel that there's two ways of trying to mitigate the risk. Namely, the first one is trying to lower the hazard, as just been presented by Mustafa. But if that fails, or if that's not possible, we also would try and lower the exposure, and ultimately do the same, also try to lower the risk. And basically, this is not just my opinion, but also a lot of toxicologists, and one that lived a long time ago already expressed that basically every material, every chemical is poisonous, but it depends on the dose whether it's a danger to your health. So next slide, please. So of course we have to look at the hierarchy of control, and I would definitely plead for trying to mitigate the hazards of the material as much as we can. So try to eliminate or substitute the hazards from occurring. So trying to change the lion into a harmless kitten. And that's what we call safe by material design, as just presented by Mustafa. But as I said, if that's not possible, or whether if we, for instance, need these materials for an energy transition, or used in pharmaceuticals or cosmetics or any other products that we really need, then there's still an option to lower the exposure levels and try to implement process designs which will also lower the exposure and hence the risk. And that's what we call safe by process design. Then in the hierarchy of control, there's the last resort that can be used to ultimately save the human being from being exposed, which is called personal preventive equipment. But that's not to be considered today, because that's really not a by design issue, but that's more like a last resort to save people. And as already presented by Carlos in his opening presentation, therefore we use in the SbD4Nano project both toxicological properties as well as exposure properties, and my presentation will be solely focused on exposure. Next slide, please. So in this SbD4Nano project, we focused on the safe by process design in this work package. So what we did in this, and what I will present today, is the collation of existing and newly generated data on dustiness release and exposure throughout the full life cycle of a product's use. Then, based on these data, we developed a fit-for-purpose nano-specific exposure model to do an assessment of your exposure for a current situation. Then we developed a safe by process design specific module to help the user in selecting the right mitigation strategy. And together with that, also already expressed by Carlos in the opening presentation, is a cost analysis to also enable and facilitate the user in making the most cost-benefit decision. And of course, I'm presenting here the result of it. This was a collaborative effort of a lot of partners in the project. Next slide, please. So as I said, we started off with a collation of existing and newly generated data. This was a combination of existing data, so the information that was already available on release and exposure of a wide variety of different nanomaterials in a wide variety of different activities, scenarios, and processes. And together with that, we also harvested from our stakeholders within the project, but also outside the project, the needs for additional risk management measures or exposure measurements or exposure controls. And I will not go into the details of all of these figures, but you see that it was quite an extensive work. And if you want to know more, please visit our website or read the deliverables of our project. Next slide, please. More importantly, based on these data gap analysis, we were able to identify the gaps in exposure data that we knew of, and we were able to fill these data gaps. So within Itena, they had a test chamber in which we could mimic exposure situations, perform measurements, feed these measurements into the eNanoMapper database, use a lot of these data together with the existing data to build an exposure a quantifier model, and then based on that build a computerized IT tool which can be used to assess your exposure. Next slide, please. So as I said, we in totally harvested 131 individual exposure measurements. We fed these data into to an exposure template that was developed in a previous project called Gracious. We use different measurement instruments to measure the exposure levels, which all measure different size ranges and all different characterizations of the particles in the air. And based on that, we built a conceptual model to assess the exposure based on determinants that we feel influence the exposure level. Then we performed the statistical analysis called the Bayesian modeling and machine learning models to identify exposure multipliers in addition to a baseline task exposure to derive as an output a quantitative measure of exposure to nanomaterials during a certain scenario and activity. As you can see in the right down corner, we were also able to identify the uncertainty around that estimate by implementing a 95th% confidence interval. Next slide, please. Then, of course, to enable users to actually use this model, we built an IT tool based on different activities, different questions that were answered. The user is able to generate an exposure estimate for the current situation and compare that to a hypothetical situation that's based on safe by design alterations. So you can actually make a modification to your process, and then in the comparison page you can see what the influence of that change in your exposure scenario or your process will do to the exposure estimate for that scenario. Next slide, please. And this will actually facilitate first of all a good fit to the exposure data. So it's not, let's say, only by expert judgment, but also underpinned with measured data, as I just explained. There's sufficient explanation and guidance provided in the tool to guide users to making the right SbD selections. There's visualization of the results to make a comparison between different scenarios. And we wrote a scientific publication about this if you want to know more about the scientific details that underpin this model. And more importantly, and that will be shown in the next presentation by Ralph, is that this model is built into the e-infrastructure that was the final result of the SbD4Nano project. Next slide, please. So in addition to this exposure assessment tool, which assesses the exposure for a certain scenario, for a certain nanomaterial, we also identified and made a release hotspot scan, which helps the users by asking a few simple questions of creating an overview diagram of all the releases during the life cycle of that specific product. The user can make use of default values that are derived from the ECHA SPERC, so the environmental release categories, but users can also enter their own findings and then make let's say, user-friendly and a fit-for-purpose assessment. There's also a database with release factors underneath that, so users can actually select from that database to use that, and then ultimately there's a dynamically built-up visualization of the workflow, which is presented in the next slide. So the end result of this release hotspot scan is identifying all the different releases throughout your production phase, use phase, and end of life phase, and therefore you get a nice overview of where the most of your release in your process takes place, and therefore it also gives you a good indication where you can actually mitigate or manage these releases, so to lower the release to either environmental compartments or indoor air compartments, which may cause human exposure. This tool is freely available on our Diamonds web platform of TNO, but it's also made available in the e-infrastructure that will be shown in a minute. Next slide, please. No, this was just to indicate what we did. In addition to inhalation exposure, there's not so much research done on dermal exposure modeling of nanomaterials, and therefore we felt that in addition to the inhalation exposure model that I just presented, we also like to give some sort of a decision tree based on simple questions, whether dermal exposure is an issue for your situation and whether you need to actually assess dermal exposure. So it will not provide you with a quantitative estimate of dermal exposure as you would get for the inhalation exposure, but it gives you an indication whether you should consider dermal exposure in addition to inhalation exposure. Also, this tool, and this is still under development, but will be made available soon, definitely before the end of the project, and it will also be added to the e-infrastructure so that people can use it in the SbD4Nano e-infrastructure. Next slide, please. So then what I told you up till now is the exposure assessment part. So analyzing the exposure of the current situation, but of course, as I started off with, you would like to make changes to your process to lower your exposure, reduce your exposure, and manage the exposure. And therefore, in addition to the NEQ model, so the nano exposure quantifier on the left-hand side, we added the Safe by Design module that comprises of several factors and components that I will show you in the coming slides. So first of all, we analyzed the risk management measures effectiveness using the exposure control efficacy library. We had Itena had a computational fluid dynamic model implemented and used, which can also be used by the users of the system. Then we made e-cards that was mainly done by CEA as a partner in providing guidance to the users on which risk management measures they can use to lower the exposure. And then finally, we had an exposure directionality assessment to also guide the users for the right SbD strategy. Next slide, please. So first of all, the e-cards. So the e-cards are meant to provide information on different risk management measures to take in the hierarchy of control that I started off this presentation with. So we identified 9 risk management measures and 13 different subcategories, which are listed on this slide. And in the next slide, you will see which information is comprising of these different e-cards. So these 22 e-cards in total have the structure, as you can see on the left-hand side. So they have a definition or a text of what the risk management measure is. In this case, it's about local ventilation system. So there's a description of what it is. There's an explanation of which level of hierarchy of control it's in, and there's suggestions for best practices that can be used in using local ventilation system. Then there are links to relevant documents, and there's a set of criteria which are identified at the middle of this slide. First of all is the complexity of the implementation of these risk management measures, so whether they are ready to use, whether there's additional development required, or whether they use maintenance, etc. Then, of course, the effectiveness, and you can already see that this is a bandwidth of the different effectiveness values. So it's not just one value, but based on how you use it or how you implement it, the effectiveness can change. Then the target group for which the management measure is implemented. So is it for workers, for consumers, or to protect the environment? Then the lifetime of the risk management measure. So is it for single use, like a mouthpiece or face mask, or whether you can use it for multiple years or over the full process lifetime. And then, as already was indicated by Carlos, there's also the cost assessment, because of course a full-blown ventilation system, as you can see on the left-hand side, will cost you more to implement than just handing out face masks. And if you want to know more, there was a presentation given at the NanoSafe conference this year, and we did this in collaboration with the Sabina project. Next slide, please. So this is what the e-cards look like in more detail. So you find all the information that I've just told you. So on the left-hand side, you see the overall e-cards for local ventilation system, and on the right-hand side, you see the e-cards for the enclosing hoods, which is just one type of local ventilation system that can be used. So if you want to know more, all of these e-cards are available on our Diamonds platform or in the e-infrastructure that will be presented by Rolf later. Next slide, please. So then, as I mentioned, Itena developed a computational fluid dynamic model, which was based on a definition of parameters or variables that were used. Then they generated experimental data, used mathematical models to build up the CFD model that you can see a visualization of on the left-hand side. And this model was validated by using the case studies in the work package 6, so the case study development in our project. And then, of course, in the end, it was also implemented in the e-infrastructure. And if you click once more, Stella, you will see let's say, a screenshot of what it currently looks like, and people can look it up on the website and use it. Next slide, please. And then finally, the cost analysis was done, and it was done in coherence with the work that Mustafa just showed, because it's not just the costs for the exposure management measures, but also, of course, to the risk management measures to lower the hazard. And this all led to a cost-integrated economic scoring system, which is implemented in the tool and which will be shown in a minute. Next slide, please. So to summarize what we've done in trying to save by process design, so to lower the exposure, we started off with identifying data gaps and harvest all the information that was available to try and lower exposure. We did experimental work to fill these data gaps and to provide workplace studies to measure exposure. Based on all of these data, knowledge, and expert judgment, we build an exposure-driven modeling framework that allows the users to assess the exposure for their situation. If you want to know more about the release hotspots of your product throughout the full product value chain, you can use the hotspot scan. And if you feel that in addition to inhalation exposure, dermal exposure may also be of an issue, we are developing, which is almost finalized, the SabiDerm module that's added to the exposure model. And then, of course, the most important part is the nano-specific safe by process design module, as integrated in the e-infrastructure, which either gives you information by the e-cards on which risk management measures you can use, the effectiveness of the exposure control measures using the library that we have, the CFD model that I just showed, together with the cost estimation, and hopefully all of this information will guide the user towards making the right decision in trying to lower the exposure to nanomaterials. And I think this was my last slide. Thank you very much for your attention, and if there's any questions, I'm happy to answer them or to answer them in the breakout session that we have later on. Thank you.
Great. Thank you very much, Wouter, for this presentation with lots of information and different bits and pieces about safe by process design. There is no urgent question in the chat, so I think we can also, looking at the time, move on with the next presentation and address any questions or comments in the Q&A session later. So I'm handing over now to the next presenter, Ralph van Houten from ThinkWorks. And he will give a live demo of an industrial example with the SbD4Nano e-infrastructure. So now we are all curious how these strategies for safe by material design and safe by process design come together in the e-infrastructure. So, yeah, Ralph, thank you.
Thank you, Suzanne, for this nice introduction. Also, I would like to thank Wouter and Mustafa to already introduce a bit the highlights of safe by design from product and process design, which we will see how these come together in the infrastructure. I hope you can see the presentation at least. My name is Ralph van Houten from ThinkWorks, and ThinkWorks is responsible for the development of the implementation of the infrastructure. And I hope in the next half hour to introduce you, based on an industrial example within our case, how the infrastructure can be used to support safe by design and safe by design strategies. Before I go to the live demo, I just first want to briefly introduce you on the case itself. It's one of our industrial case partners, Creative Nano, who use titanium dioxide nanoparticles in paints, and they apply them for their photocatalytic degradation properties. So basically, you can see here in the middle a schematic overview of the nanoparticle, which under light, normally under UV light, can create reactive oxygen species, and with these species, pollutants in the air are reduced. But of course, what our partner wanted to achieve is that this paint can also be used in an indoor scenario where less UV light is available. And in order to make that happen, sulfur doping was added to these particles in order to to reduce the band gap. And that basically allowed these nanoparticles also to become photoreactive indoor. And of course, that's the first step in the design process here. As of now, I will call these sulfur-doped titanium dioxide nanoparticles the baseline nanoform. So this is a starting point from where the Safe by Design strategy started within the SbD4Nano project. And actually within the project, our partner Creative Nano has made surface modifications to the particles by adding silica layers around it, and we will see later in the tool how that manifests. So this is a bit about the context. Of course, what's also very relevant in such a scenario is to have an understanding about the different use scenarios throughout the life cycle. And you can see here some examples where, of course, in the early stages of the product design, you have to synthesize the nanoform. This is a sol gel synthesis for these particular particles. These are then dried and then ball milled, as you can see here in ball milling, and then it's put in an oven where a calcination step is done, and after that it's ball milled again, and then you have here in the bottle you see the actual nanoparticles being shown. These are then put into a formulation and end up in the waterborne paint, and the paint is then applied indoor in the compartment where it has to perform the photocatalytic activity of reducing air pollutants. So this is a bit the contextual background of the case I want to use as an example in the infrastructure. So let's now go to the infrastructure itself. So the infrastructure, as already mentioned earlier, it's an online tool. So at some point you need to ask for credentials once it is become available, and you then need to log in. So let me show how that's done. For the logging in step, and then of course just provide a name and a password, and then you get to the point, basically to the landing page of your case. So let's go here, and this is in line with what Carlos was showing earlier, where at the top you have certain groups where members can cooperate together in a group or in cases. And at the bottom you can see the different resources available. And just to give you an impression, here you can see the different resources already presented earlier. So there's a quite extensive list of all different kinds of resources in the community available. You can apply filters on that and to see what potential resource could be become available for your Safe by Design ideas. Another part of the resources is this search for relevant documents. So in this case, I could be, for instance, be interested in titanium dioxide, band gap, and dope. And then we can see what articles and papers are out there. And you can see there's quite a set on that. And you can see here an overview of the most relevant and most recent articles which are related to these terminologies. So that's very handy if you want to get more insight on background information on your products. Another part of the resources available is the link to the eNanoMapper database, where you can find a collection of physicochemical properties and tox and ecotox related data for reference materials, and also materials made available from earlier projects, EU projects. There is a tool in here for the regulatory, to identify the regulatory documents and guidelines which might be of application for your nanoform or for your nano application. And you can also see here, as already mentioned earlier, the different resources in the TNO Diamonds platform, like the e-cards, the exposure quantifier, the hotspot scan, the computational fluid dynamics, and A-cell and the dermal exposure assessment. And we will see later in the assessment itself how we make use of these resources via the API. So let's now go to the actual case. So the starting point of using the infrastructure is basically when you can start a new case, of course, a safe by design case. But of course, for the preparation of this, I already made a case due to limitations of time, of course, we have. And when you enter such a case, you get to this part of basically the landing page of your case. You can provide some background on it, provided a name, and then you can see here the big categories. So you have to introduce the products along your life cycle, the life cycle description itself. Then you can go to the safe by design suggestions, the ranking, and the comparison. There is not a strict order in which you have to follow, so it's more a modeling environment. So you can start with, for instance, describe your life cycle first and then introduce the products as you go. But you can also do it the other way around. And for now, let's have a look at the introduction of products and materials. And you can see here on the left a number of products and materials which are relevant in the life cycle of this case. And you can add as many as you would like. So you can introduce new nanoforms, substances, mixtures, and even articles. And in this case, I introduced some precursors, raw materials used for the synthesis. So you can see here the ethanol, the water, which is actually made by the industrial partner itself, but also intermediate process products, and of course the nanoform itself. So in this case, it's the baseline nanoform. It's the sulfur, the titanium dioxide nanoparticles with the sulfur doping inside. And if you look, you have then to provide some information on to identify it. So provided the name, the type, what is the origin, potentially any descriptors, and then the tool asks you to provide basic information on the nanoform. So what is the composition? You can see the core is made of the titanium dioxide, and the dope is added as sulfur constituent. You can indicate the layout, so which of the constituents are first getting in contact with the surrounding medium. Crystallinity, of course. In this case, it's anatase, the titanium dioxide. The shape of the particle is mainly spheroidal. You can the size information on the primary particle. In this case, it's roughly 11 nanometers, and you can also provide information on the surface area. But as we already saw earlier in the presentation of Mustafa and also in Wouter's presentation, there might already be certain assays conducted for this nanoform, like Fischem characterization. In this case, there is already dustiness measurements has been conducted with the vortex shaker. So that information can also via templates be added to enrich your case. But you can also see here that ecotox data is already added for this nanoform. So here, this is a freshwater algae cyanobacter, but also you can here see an other Daphnia related ecotox test. So all that information can be added if that information is already available for the baseline nanoform. Of course, the nanoform is also to be used in the context of the paint. So you can also add in the product list the paint. In this case, the paint base. I modeled it. It's basically the paint without the nanoform in it, and you can see here the different constituents. So the dispersants, the thickener, the stabilizers, the pigment, and the fillers. And then I combined this paint with the nanoform, and you can see here the composition that the nano-enabled paint is the paint base together with the particles. So there's 5% of the particles put into the paint. So, and this is the way how you can model basically all the different materials and nanoforms in your product, relevant in your case. If you provide information, you can also analyze it at the top. So you can look at the different properties which have been entered, and you can already see I also introduced the safe by design variation. which is the one with the silica layer around it, and I also provided all the information on that. And then you can, for instance, look at the plots. And in this case, I looked at the ecotoxicity, compared the original baseline nanoform with the silanized nanoform. You can clearly see that the original form is more toxic than the one with the silica layer around it. If you go back to the products overview, you can see here the titanium dioxide, basically the Safe by Design version, and I also added the TEM page, the TEM image here, and you can clearly see the silica layer around the titanium dioxide, and also these particles are a bit smaller. So basically, what the tool here offers is the ability to add materials, add precursors, introduce articles, and all the relevant products along the life cycle. And you can do that together also with downstream users. The next step would also be to introduce the use scenarios along your life cycle. And of course, that depends a bit on what you're interested in. You can focus on just the manufacturing stage, but you can also consider the use phase or even the end of life if that is relevant for your case. So here you can see I've introduced 4 different use scenarios. So the manufacturing of the baseline nanoform, so without the silica layer. The same, but then with the silica layer, the paint formulation, so the weighing and the mixing of it, and then during the service life when the paint is applied on the wall. And when we now go to the more detail of this use scenario, but because each of the use scenarios has contributing scenarios in it. And if you remember the slide I showed earlier, where you have these different process steps, basically these are represented here. So you can see the sol gel synthesis step, the drying, the ball milling, the weighing, the calcination, and then the second ball milling step. And if we go to the sol gel synthesis, you have to first identify what exactly the step is about. So identify the category of the activity and the task itself. You can provide some background information on it. And then you have to introduce the inputs and the outputs. So basically the mass flow and the materials involved in it. So you can then select just from the materials you introduced in the overview, in the product overview, you can select them there. If they're not in there, you can add them. You can also provide information on the prices. So how much does it cost per volume or per kilogram, which is then later used in the cost calculations. And also you have to relate how much of the input is required to get to the output of this activity. And by linking the different activities within this use scenario, so the outputs of the sol gel synthesis with the drying and the ball milling, etc., then by linking all it, the tool can then at the end, if you go to the output and identify that we want to produce 5 grams of the nanopowder, it can then calculate the amounts of materials involved along the entire life cycle. And in this case, within this use scenario. Let's go to the paint formulation part, because here I introduced 2 activity steps, which is the weighing. And you can see here a picture of the weighing, which is actually done within the fume hood, and you can see the scooping of the nanopowder. And then the second step here is the paint formulation. You can see here this is the lab scale formulation, so they also do it in bigger volumes, and then that's basically in this equipment. But for now we look at the lab scale. And we are interested in potential exposure and release scenarios here. So for the weighing, you can introduce, and you already saw that, a potential in the picture, that the potential particles might be emitted here. So you can introduce a potential release, and connect that release with the release material form, and also the zone where that release might go into. And in this case, that's the fume hood, which is in that compartment. So let's have a look at the compartments. The tool, besides the use scenario, also allows the introduction of different compartments, like the synthesis lab here. You can see a picture of it. So this is the entire room where the activity takes place. You can see here the fume hood, So you have to provide some dimensions of this room, and you also have to subdivide the room into zones. For instance, the zone, the floor is a zone. The far field is the biggest volume of the room where the activities are not happening directly, but also we have the near fields like the fume hood. So there's a small zone here, and also one surrounding the fume hood. And there is one where the ball milling takes place, but that's located behind this desk. So basically you have to subdivide the compartment into zones where certain activities might release their particles in. And you can see here, so within the fume hood, we have the weighing and the formulation of the paint. Okay, we also can indicate what risk mitigation measures are in place, like this enclosing hoods, the fume cupboard you already saw in the picture here. And the tool can use then the e-card to give some feedback on what kind of controls are potentially available, what the effectiveness is. So this is basically already mentioned by Wouter earlier, but you can then identify if the control selected is actually the one mentioned here. Let's go back. You can also indicate costs and energy consumption involved for this risk mitigating measure. So you can do that for all the other rooms as well, if that's relevant. So there you can see here the room where the furnace oven is located, and also the outside compartment here. So basically, this is the air compartment nearby the vicinity of the building. So let's go back to our use scenario. So that's the paint formulation, the weighing. Like this. So now we have introduced our release and our emission, and the next step is that we can now need to identify what potential hotspots of exposure there are. And as you can see on the picture here in the contributing activity, as you can see in the picture, you can see that there might be potential dermal contact within the fume hood. The hands are located here, but the breathing zone is outside the fume hood. And that's actually modeled by indicating the two different pathways, one the inhalation in the operator zone and the other one in the fume hood. You can also indicate if certain protective equipment is already in place, which ones, and if they are actually applied. And you can do this for all the activities along the life cycle. So once you have done this, and I also did this for the paint formulation, you can see there's also one hotspot here. Then you can go to the overview of the hotspots. And here what you can see is the different activities along the life cycle. And I focus now for this example on potential exposure scenarios and hotspots due to the weighing and paint formulation step. So there might be also here, but I didn't, for this example, didn't identify them. And you can now go to identify the exposures which might occur at this point. So let's click on this, and then we go to the exposure assessment for this this scenario. And here you can see that I used the NEQ model, which was earlier presented by Wouter. And the model basically passes the information I already provided to the NEQ model. So it basically answers these questions the NEQ model needs. And then we can retrieve the results back from the NEQ model, and these are then provided here. And as you saw, there were also PPE used. So then you can account for the PPE effect, and then this is the actual dose, the inhalable dose for this scenario. And you can also do that for the other, for dermal and other inhalation scenarios. Let's go back to the hotspots overview, because here now you saw how you can provide information. You can also do that by providing measurement data or using other models. But the next step would be that these hotspots introduce safe by design goals. You can see here these pentagons. These are safe by design goals introduced for in these different activities. So for the weighing, we have a safe by design goal, and also for the paint formulation, we have a safe by design goal. But we have also, you can see at the last column, a performance goal, and that's related to the last activity in our use scenario. That's the surface life of the paint, because that's actually when the paint is on the wall, then it has to perform its actual function. So here we make the function operational and say, okay, this is the point in our life cycle where the function matters. Okay, let's go back to the hotspot. So now let's have a look. We have now introduced the SbD goals, but now what kind of safe by design strategies can we apply to reach these goals? So let's click on this pentagon here. And as you can see, for our weighing scenario, the tool creates automatically a so-called influence diagram. And at the farmost right, you can see here the pentagon saying lab worker health is our goal, and we want to maintain it or increase it, improve it. And now you can see, based on the hotspots, that there are two potential risks we should mitigate: the inhalation risk, and here the potentially dermal risk. So let's focus on the inhalation risk. Inhalation risk has two components, as already mentioned earlier. You have the exposure, which you could try to mitigate on the one hand, but you can also try to mitigate the hazard of the nanoform itself. And you can see that the tool has hypotheses, and these hypotheses allow the tool to create automatically causal relationships between these different features as they go along to the right. So you can see here, for the hazard limit, we want to increase that. The higher the hazard limit is, the lower the risk. And we want to do that in relationship to our exposure, which is the route of exposure inhalation. So the lung organ is our targeted subject. Within that, we have certain cell types. We already saw earlier that the cell types, like the macrophages, are the ones relevant in this region. And these macrophage cell lines might be inflicted by oxidative stress, which might be caused by ROS generation, which might be the consequence of the surface chemistry of our original particle. And the tool automatically knows, based on the information we provided, that sulfur and titanium dioxide are ingredients. And as said, as Mustafa already indicated, the sulfur concentration might have an influence of the surface chemistry, might influence the oxidative stress, etc., and cause in this branch the hazard. When we focus on the exposure part, we want to mitigate or perhaps reduce the potential uptake. Now, one of the strategies could be to use a filter mask, but as already said, that's the last resort. So we try to mitigate the concentration, basically the exposure of the workers in that room. That can be done by limiting the presence of that worker in the room, and you could optimize your weighing scenario, but you can also try to lower the concentration nearby the fume hood, which you can do by perhaps moving it to a larger room. The dispersion will then lower the concentration as well. You can try to further increase the ventilation rate, but you can also try to mitigate the transport from the fume hood into the near field of that fume hood. And that you can do by try to lower the concentration within the fume hood. And that can be done by enclosure of the weighing, or perhaps try to avoid too drastic scooping of the weighing, so not to put much energy in there. But you could also think of try to lower the dustiness of the particles, which you can do by either modification of the size, by reducing or increasing its aerodynamic diameter, which make the particles settle faster. But another option could be to try to have these particles coagulate faster, so that they will also settle earlier. And that you can do by changing the chemistry or the layout of the particle. But you can also try to influence that by changing the moisture content. And the moisture content of the powder, in this case, you could influence by trying to change the relative humidity inside the fume hood. Or you could also try to add a surface modification to your particle, which make them coagulate more fast. This is, of course, from exposure perspective. It's not sure that that's also the best strategy from the product performance strategy. So let's have a look what the tool can, how the tool can use its network to guide the user to potential scenarios. So you can see here an overview used, extracted from the influence diagram. It says, oh, you could try to increase the particle size a bit because that might lower the dustiness and as a consequence lower final exposure to the workers. But you could also think of, let's have a look here. Let's have a look if we have a nice one. You can lower the product amount of the weighing. You could increase the automation level. As said, you can, let's have a look. If there are other, you can add a hydrophilic group, but as you can see here, the tool also noticed that there might be opposite conflicts or that there might be a local optimum. So by adding more hydrophilic groups, it might have an opposite effect on another pathway in that influence diagram. And you can see at the far right that it tries to use these conflicts, etc., to give it a first score for this particular goal. Of course, this is from exposure perspective and from hazard perspective, but as I said earlier, we should also consider product performance. And let's have a look at that. So if we go to our hotspots and look at the performance part, then a similar influence diagram is created. But this time the goal is to maintain or increase the performance of our paint. So the photochemical reactivity here, and that's actually provided by the ingredient, which is the nanoform. And there are different pathways which can contribute to this ingredient by perhaps make sure that there's more light available to make it more operational. Make sure that the nanoparticles within the paint are well dispersed so that they are accessible. Look at the performance of the ingredient, the nanoparticle itself, or perhaps the availability of the nanoparticles within the paint. So the amount, the loading within the paint. And here, if you look to the performance of the individual nanoparticle, you can see that photon absorption, if we can promote the photon absorption, it will increase the photocatalytic activity. And we should try to avoid the charge recombination rate, which basically neutralizes the excited state. And this actually, you want to avoid this and you want to promote this. And then you can see how, for instance, the change in surface area can contribute to higher surface area, will also promote more photons to be absorbed. You can see hypothesis related to the band gap, how a dopant can influence the charge recombination rate, but also the band gap. So here you can also have conflicting scenarios. So if we go to the potential strategies to increase the performance, you can see that reduction of your size might be an option. And actually, if you remember this, the addition of the silica, actually it produced smaller particles. So by the addition of that silica layer, the particles actually became smaller. You can also think of the loading, put more of the particle in your paint, but that makes it more expensive, of course. And there are other strategies you can follow. So basically, the tool helps the user to think of different potential strategies to follow. Okay, but that now we have seen how the tool can help the user for individual goals. But of course, if you go along your life cycle, you have to take into account all the goals together. So you can see here at the proposed SbD strategy at the top that we have these 3 goals, and now combined, you can see how they all contribute to the overall SbD performance. So in this case, because we have 2 different activities, which both are conducted within the fume hood. So making adjustments or modifications or improvements to the fume hood has a beneficial effect on both the activities, and it doesn't impact the performance of your product. And as you go to the bottom, you can see if you try to change modify the nanoparticles itself, then you might get into conflicting situations that what is good for the performance might have an adverse effect on the exposure. So you can clearly see that the strategies to go for here would be, in this scenario, would be more application of the risk mitigating strategies, local controls, and perhaps also on the activity itself. But of course, if you go along the life cycle, more goals might be introduced, and this matrix might become more complex. And then finally, at the top, we have the comparison tool, and this allows to make comparison configuration. So you can configure different scenarios. So this is, for instance, the baseline scenario, and I also introduced the SbD modification scenario, and I want to look at them from a performance and cost perspective. For the baseline, I selected the manufacturing activities for the baseline, and for our Safe by Design version, I selected the other activities which are used to synthesize the modified version. And then I can go to the compare scenarios, and here we will link to what Wouter was showing earlier. We will go to the performance, and Mustafa already showed that measurements of the performance of the baseline scenario and the SbD modification, you can see here the differences. So there is actually an increase in the performance and a reduction in the hazard, and you can also see here the cost comparison. So this is the baseline, the input materials used, the energy involved, and I didn't enter all the labor costs, but you can provide the information as well. And you can see that the Safe by Design strategy hardly is more expensive than the original baseline. So from a money perspective and time perspective, there is not that much of a difference. So you can use these kind of considerations from tox perspective, exposure perspective, performance and costs to make a decision more in line with the knowledge coming from the tool. Of course, the idea of the tool is to help you reduce the number of trials and errors to get from your baseline scenario to a more safer product. And this basically is a rough overview of the tool. There's of course much more in it, but for now, I hope this gives at least an impression what the infrastructure can offer you as a Safe by Design tool, and to help SMEs and companies to at least increase the speed and the efficiency of Safe by Design processes. Thank you for your attention.